has a PhD from Cornell University, which she obtained in maize genetics in 2006. She then went on to do more postdoctoral work also on maize genetics at Cornell, and then went to industry for several years as a research scientist and project manager for the Nature Source Genetics company. She's been with Breeding Insights since January of 2019, and she will talk to us today about the concept of the Breeding Insight platform and how it's relevant to ag biodata. So with that, Moira, go ahead and take it away. Thank you.
Okay, I think I've found the unmute button. So I want to thank you, Monica, and also Jacqueline for the invitation to speak to everyone today. Just looking through the list of the people on the call, there's a lot of new names for me, so this is an excellent chance for me to kind of talk to you about what I'm doing. And I also need to thank Jacqueline for her excellent coordination of the seminar series. She was very helpful in providing what kinds of backgrounds everybody might be coming from. So I'll do my best to try to reach everybody's into their background just a little bit, but you're going to probably find this is not typically completely focused on databases. So I hope that there'll be something new for everybody here. So as Monica mentioned, I'm the director of Breeding Insight, and my talk today is really talking about how do we empower specialty breeders with integrated tools that allow them to do genomics, phenomics, and data analytics in a single unified platform. And we are an ARS-funded mission, so our goal overall is to transform breeding by enabling the implementation of genomic insight and selection as part of routine breeding programs across all of the ARS. Before I go on, I should have mentioned I'm a very fast talker, so if I'm going too fast, please just do use the go slower ping on the Zoom call, and I'll slow my speech down. Okay, so when I think about what makes private sector breeding programs so incredibly effective at releasing new varieties and pushing genetic gains further and further, it really comes down to a couple of big pillars. First is they have rapid in-house genotyping. They genotype just the markers they need for their application, and they get those results back in about 3 weeks. They also have really robust phenotyping methodologies. They have tractor-based sensors. They have iPads in every hand of any worker in the field, and they're able to use other kinds of standalone phenotyping tools like laser measuring for height. They also have a whole department that is able to help breeders use these tools, but also to take the data that comes out of those tools and to use them in their analytics. So they have biometry departments that help them with their field layouts and randomization and statistical testing afterwards. And it also is responsible for developing new technologies that they can roll out to their breeders. And lastly, big companies are very good at coordinating these activities. They provide expert people that to work the greenhouses, to know how to run the field sites, to ensure that the tractors are maintained. They have all these things in place that most public sector programs do not have. They don't have this tight coordination of all of the activities they need to do under one umbrella. So what we at Breeding Insight are really trying to do is to bring these capabilities to the specialty breeders within the USDA with an economy of scale. So if we can build the resources up from within Breeding Insight and then populate it back out, we're able to start hitting some of these key pillars and bringing those services right to the breeders that are trying to modernize their program or start using genomic selection. And they need to have all of these things somewhat in place before they can really effectively move forward in their genetic gains. So how do we build Breeding Insight? So we're starting with 5 pilot species, and I'm just showing them here on the right side, and I'll talk a little bit more about that in the next slide, but our basic challenges here are we have a variety of different types of species here, and we didn't know a lot about any of these when we first started. So we had to go and meet with each of our ARS breeders to really understand what their facilities look like, what their computer systems look like, how was their Wi-Fi, what is their breeding schema, how much land do they have to manage in cultivation. So we took the time to visit over the last 6 months each of these programs and stay for 2 days to really integrate ourselves into their program and understand what their needs and wants are. And so if some of these crops are familiar to you, the next slide's probably going to be a little bit of a review. But if you're not familiar with even one of these, you might find some surprises here. So we have a lot of challenges, and these crops were chosen for a number of different reasons. And one of them is that they offer a chance to tackle technical challenges, biological challenges, even logistical challenges of the breeding cycles. So in the case of blueberry, this is an auto-tetraploid. Right now, there's very few markers available, only a few SSR markers. It has a very long cycle time. If you're talking to a breeder and they say, we don't even look at yield data until year 4 of the age of these bushes, they're just not, it's not predictive before that point. And when we spoke with our breeders, there was a lack of quantitative data. It was a lot of, I like this, I don't like this, but I don't really know why I like this or why I don't like this. So we have a bit of an uphill battle there, getting them to start thinking about how do they quantify their selection decisions. In alfalfa, it's another autotetraploid. Again, because of that nature, it has very few markers available. Also, the cultivation system for alfalfa requires multiple harvests for years, and typically a cultivar will stay in the field for multiple years. So 3 to maybe 5 years, these plants will stay on the same land without any additional cultivation, except for maybe some additional fertilizer, if that. But again, what we find with this is that there is also a lack of quantitative data in the breeders' historical data files. Apparently, they even, when they look at scaled traits, sometimes they're looking from 1 to 5, and then they come back the next day and they say, well, I want to change my scale from 1 to 9. And so you can imagine that this is a real difficult problem when you're trying to establish a trait ontology that doesn't change from day to day or field to field. In salmonid fishes, we're very lucky we have a diploid, so it's also good to have a little bit of variety there. There are a lot of markers available, but the cost of genotyping is very high. For the trout program, it's actually $50 a sample. So it's just exorbitant costs to actually phenotype your population. And because 80% of the traits are lethal, you're not actually able to use the parents, you're not able to use as parents anything that you collect your lethal phenotypes on. So fillet yield or body weight. So you have to do selection basically on your SIB populations in your breeding pool. And also we found that the data collection is not really digitized yet. It's still mostly being done on pen and paper. That includes metadata collection on animal welfare, which is another reason for us to kind of work with them, is to try to help them with their animal welfare management and to modernize that so that it is a more active system instead of just a pen and paper with no, clear direction of who should be taking care of salinity if it drops. We also have sweet potato, which is an autohexaploid. There are very few markers available, but there are some, there's a good community resource out there that's looking to stabilize a marker system and develop those markers. It is highly heterozygous though, so whether or not that marker system is going to meet the needs to call dosage and also phase those haplotypes remains as an open question. We're trying to help them with that decision by guiding them towards technologies we feel are more reliable, robust, have good service contracts with third-party vendors, but will also give us the necessary depth to use that dosage information appropriately. Also, there's not a lot of interest right now in confirming pedigrees, and I think that for a polycross plant like this is a real problem, because there's a good chance that you have disparate mixes of populations within what you consider your breeding pool. And it would be ideal to have some pedigree information that you could confirm as to who were the males for these individual clones. And also, pretty much every time you make a biparental cross in sweet potato, you basically get a whole population that looks like a diversity population. So when they see something they like, they just clone it. So they don't know a lot about what's underlying in the genetics that's giving them things that they like, because they're not, they're starting to quantify their phenotypes, but they don't have any genotypes to really map it back to. We also have table grapes. So this is a small segment of a large market for grape. We have wine grapes, raisins, etc. But we're really focused on the table grape program at the ARS. This also is a diploid, and they're a little bit ahead of the rest of our groups, as they've already settled on a marker technology. They're using 2000 Rh-AmpSeq markers from IDT. Again, it is highly heterozygous, but the way they've designed their marker system allows us to call those hets with very high reliance on the data, because they are, each of these markers is actually a collection of SNPs or indels within a locus. So we can actually look at local haplotypes in grape. But when you're working in pre-breeding, you're working with vines that are either male or female, so that can be problematic in your breeding program. And also in table grapes, we want seedless products, but it's hard to also breed if you're always producing seedless things to get back to a state where you can rebreed it with that as a parent. So these are just some of the logistical, the technical challenges, the biological challenges of the first 5 pilot species within our breeding insight program. So after we meet with all of our groups and we really try to understand as deeply as possible their needs and wants and what's going on in their program and how it's currently run, we have resources to help them set up their genotyping platforms and to manage that data. And we also are helping them migrate legacy systems or Excel files for phenotype data into data management resources. And the last part is we're working to develop a software platform, considering what publicly available components are already out there. We consider what is functionally needed for our breeder specified use cases. And from that last slide, you might get an idea of there's quite a lot of things that most of the database programs aren't able to handle very well from the get-go. So we need to make sure that what programs we do use, either we work very closely with those groups to ensure those functionalities get a priority for improvement, or we find another platform component to work with that already has that. And because the components that are currently out there in the public sphere are really kind of one-off pieces of software, they don't really interoperate at all right now. So we're working on using Rapi, which I'll talk a little bit more about, but I think this group is probably pretty aware of what Rapi is, to make a seamless back-end communication between these different components. And then we are also building a web service layer that will go over top of this integrated system that will be a user interface that is species specific and will be intuitive. And I'll give some examples of what I mean about that when we get further into the talk. So we like to think of the breeding cycle breeders are going through as a 12-month cycle. So whether the cycle for release is longer or shorter than that, the operations day-to-day for any given breeder is based on what you can do in your calendar year. So when you plant, when you're taking your mid-season phenotypes, when you harvest, etc. So all of that still happens on a 12-month cycle. So breeders are coming to their computers during those times to do different functions, okay? So if we look at what the digital ecosystem is and how it matches up with the breeding cycle, we have the management of stocks and pedigrees, the decision of what crosses to perform and how to manage that in the field or greenhouse or wherever those are being done, and the need to design and manage trials, including the data collection and the setup. And then we also have a period of time in the spring and summer, early fall, where it's really heavy on the capturing for phenotypes. And that could be pen and paper. Some of our breeders are maybe using voice recognition software or a tablet-based methodology. And we'll talk a little bit more about that later. And then taking that data and then having to evaluate the phenotypes, and then if they've been able to genotype at some point, they have to be able to evaluate the genotype data and bring those two things together to make decisions. So those decisions could be culling decisions, they could be additional testing decisions, or they could just be selection decisions, either through genomic selection or marker-assisted selection or whatnot. And then many of our breeders report that they have an annual report that they have to put together, and pulling all the information of what they've done over the year or past years together takes an inordinate amount of time. And if it was all the data was held in one centralized location, at least it makes it easier for the breeder to pull that forward, or even generate custom reports that get used routinely year after year. Okay. And so even though right now I'm just talking about what the plant digital ecosystem looks like, it doesn't look that different for fish, and it doesn't look that different for insects. So the basic premise is that this digital ecosystem is happening for almost every breeder that operates on a 12-month cycle, that, you know, for agriculture at least, but also for bees and for trout as well. So I've gotten a lot of questions about what is BI responsible for in terms of data, fair data management, and I kind of came up with this slide to kind of try to describe how we see ourselves. So the breeder is doing multifaceted activities in their program. A lot of them don't impact data management software, at least the way it's currently being done. There's HR things they have to do, etc. Okay, but when breeders are working with BI, we're talking about us receiving and helping them process data, helping them load and transform that data, analyze, extract results, and report and package that data up. So we expect this to be a routine process that will happen over and over again with breeder data or data from their own germplasm. And what they won't see is all the system administration underneath and any technology integration. Because of the way the interface is designed, it should all feel like a seamless system, even if it is broken down into multi-components. So BI doesn't really create any data, except for if you consider any software development as data, but it doesn't actually create any program data, but it does help store, utilize, and protect that data. And we make it easier for breeders to then push that data out to the public in whatever form that's been decided, or to do the publication of that on small subsets or whole data sets. Okay, so we're here to help steward that data forward and make sure that they meet the recommendations and requirements for the ARS's data management plan, and also to give them the freedom to make those decisions about when things go out to public, when they go into publication. That's not our decision. So I hope that makes it clear. The way our team is structured is we have a software development team. It's led by our lead developer, Tim, and we have a UI UX designer. So this is a user interface, user experience designer, and she's been fantastic in really setting out software that breeders are actually looking forward to using rather than kind of dreading a long training session for. And right now we have 3 application programmers. I'll talk a little bit more about their first success a little bit later in the talk. We'll come back to that. But also on the scientific side, we have our senior program manager, Siva, and 2 coordinators, a genomics coordinator and a bioinformatics coordinator. And they're really there as partners for the breeders during this whole adoption and transition period, and could even be collaborators long-term down the road. But the real point of them is to help guide breeders to take up new technologies, to feel comfortable with making decisions in the software, to understand what best practices are. They're really the partners of the breeders or the breeding programs. So again, this is not about data management exclusively, and we offer a lot of other services to our participating breeding programs, including helping them skim their key parents for marker development, and then coming up with an affordable and fast genotyping platform and ways to manage that data, new phenotyping technologies that are coming online. Our sweet potato breeder is using a PhenoSpecs for looking at insect predation and beetle predation damage on his tubers. So he's got the setup. It's just that the data that comes out isn't immediately usable. So we can help him take that data, push it through some machine learning algorithms, and start a training set to really help him use that and collect better phenotypic data that he can actually make selection decisions on. And the types of decision support we're hoping to provide them either right now as a one-to-one service consultation, but eventually as regularized software is for marker-assisted selection, GWAS, or genomic selection support. And our workflow-based software, we hope, is going to be a lot easier to adopt for our programs. And along with that comes that breeding consultation and curation service that we are providing to them. So I'll talk in the next couple of slides about our software components and our build strategy. So we're working on our first platform integration with a number of different programs. Some of them may be familiar to you, and I apologize if I assume that you don't know what any of these programs are, but I take that approach because I don't want anyone to feel like they don't know what these individual programs are. So there's a phenotypic database called Rebase that's run out of BTI here at Cornell. We're also working closely with the FieldLook app, which is in the Pheno apps suite of mobile apps. We'll be working closely with the Gobi project, which is genetic data management and QC and some decision support. And eventually we'll be integrating with Green Global to make sure that we really pull in those germplasm resources into this program so pre-breeders have better access to Green Global within their system, and we can port data back and forth, and it's not a big hassle to have to put it in Excel files. We want to make that all seamless. And we're connecting these by using Rapi. And so I'll talk a little bit more about the three groups on the left side of your screen in a couple closer to the end of the talk. But I do want to give a framework that this is not a system that has to look like this for every single group. So we started with these programs because Rapi, Rebase, and Gobi are all located at Cornell, so it makes it very easy for our development teams to co-develop. Okay, and a lot of these programs are quite mature. Rebase has been in active production for over 10 years, and it's continually being supported and updated. Pheno apps is already being integrated with Rebase, so and other tools. So it makes good sense to start with things that people know. And each one of these components really shares one or more of our breeding insight goals for a platform that is able to provide end-to-end support for a breeding program. Okay, so what this breeding insight, it probably looks pretty similar to some other programs you might be aware of. And so I want to give you kind of an idea of where we see ourselves in the global community. So we have our partners, which we talked about on the last slide briefly, but we also work very closely with Excellence in Breeding Platform, and we are specifically working with modules 3 and 5, module 3 for volume pricing and genotyping vendors, and module 5 for bioinformatics and software. They're using a lot of the same programs that we are as software components. And a lot of those software components came out of Bill and Melinda Gates funding. So we're trying to work closely with them so we don't replicate or duplicate efforts on our integration for interoperability. We are also working with the Feed the Future Innovation Lab for Crop Improvement from USAID. We don't participate directly in this, but they are working also on putting new tools, technologies, and methods in the hands of NARIs all over the world in key countries in Africa, Asia, and South America. And we also are working into the hands of our breeders. So these are, we have some commonalities. These are small programs at NARIs, and they're small programs at the ARS in many cases. So there may be some commonalities there that we can work together on how to solve and find better solutions. We also stay close contact with the University of Tokyo and a group called ListenField there. They are developing what is essentially breeding insight, but for the Japan NARO there. So their national program and also they would provide it to their multiple breeding companies that are really very small breeding companies. They breed for lots of different species, but not with huge programs. And they do a lot of ornamentals as well. So a lot of the crops that they're working with in Tokyo or in Japan are not crops that have been hit or targeted for improvement by any other company or program out there. Okay. So these kind of are just next few slides are just about our software architecture, just to give you a little bit of an overview. So our interface starts with our interface that is connected via Rapi to a BI web service. And we'll maintain our own database for how that web service will appear for any given program. And you can see that we've just kind of broken it down into, each species would have its own service cluster. And I'll talk more about the service cluster on the next slide. So this architecture allows us to have multi-species support, which is really important if you have a breeder that breeds for more than one crop. They want to be able to get to all of it at the same time pretty easily. It's also a modular architecture, which means it's a lot easier to swap in and out. And it also seamlessly integrates the subcomponents, and it will be Rapi compliant. And we're taking a microservice approach for our underlying component structure, and that makes it very modular. So because we're using Rapi, we can easily swap out. I'm going to try to use my pointer. Can you see my pointer moving, my arrow?
Yes, we can.
Okay, so if we are working with a group that's already using GEMS instead of Rebase, or is using a triple database, as long as it's Rapi compliant, we don't need to use Rebase. We can swap it out and use a different database system. We're trying to be as agnostic and open as possible and not make additional work where it's not needed. So if they enjoy using their triple database and they want to keep using it, as long as it's Rapi compliant, there won't be any problems with it. So this is why we wanted to do this, is because it does make every system, it can be customized to work with systems that might already be storing data in these programs that we're working with. So for example, our blueberry breeder already had something similar to a Rebase. It was a custom built database system built by OSU, so Oregon State, and they're very happy with it. So we don't want to reinvent the wheel there. We just want to make it easier for that piece of software to talk to everything else they're doing in their program. Okay. All right, the last thing I'll talk about on the architecture is our web service, which is our core development activities. And it's the connection between the user interface and its integrated components underneath. It's responsible for fetching and storing appropriate data based on user requests. And it's flexible enough to accommodate software needs into a single interface. And so it will be able to pull phenotypic or genotypic data, passport information, inventory data from a larger array of what we consider inventory. And it will also be able to call out to analytical packages to actually run genomic analyses and statistical analyses. So this flexible architecture powers an intuitive workflow-based user interface that helps breeders make better breeding decisions. So I'm going to talk more about this interface now. And it's really centered around how these breeders are interacting with their computer already in that 12-month breeding cycle. And so this workflow-based design means that we spend a lot of time with our breeders on location to understand what's being managed when and how. And we return to the breeders to ensure software that we build reflects and supports those processes. And then we can tweak it in case something is not quite right. Maybe we left out a step in our workflow and we need to create content for that. And this provides breeders with information management tools that they can use throughout their breeding cycle and throughout the calendar year without having to have multiple different places to go to do that. So we build a breeding workflow basically by following where they're moving their data around in that calendar year. So if they're designing an experiment, they're probably tapping into their inventory at some point and figuring out what pedigrees they may want to use or what crosses have already been done that they can pull forward. And then there'll be specialized other steps that come downstream. And each one of these things has specific tasks that are associated with it. Fertilization and propagation, you'll probably have need to print labels and have maps of where things are in the field, and you may have in your program a tissue culture propagation step to clone things out, and you would need to have services pulled out of a mainstream to provide that interface for you to actually manage your program better. So this is just an example of how we're building our workflows to provide context-sensitive data tracking throughout the entire breeding workflow. And so our approach really is to focus on 4 key pillars. The first is interconnectivity. So we base it on a priority on the ability to share data across software tools. We use logical task groups to help customize those breeding work cycles and program activities. And we really want this to be easy to use. A lot of the software out there right now has interfaces, but for new people starting, it's not intuitive where to start. And even if you went through a 2-week training, by the time you get around to having to do your field design, it might be 6 months later and you won't remember. It's not intuitive. So we want to make sure that our software is breeder friendly, context sensitive, and is designed with the existing breeding workflows in mind and coordinates that. And our discovery process is really dedicated to understanding each program's specific breeding processes. And it's designed for individual specialty crops that aren't using huge enterprise software or need enterprise scale software. So coming back to the logical task groups, I just want to kind of throw this out there. This is just an example of how we break down task groups for our workflows. So if you're making labels, you really only need to look at what's in the pink bubble there. You need to attach experiments, you need to do setup, what common layout you might have, and you need to print. You don't need to be looking at what's happening in a display experiment necessarily. You don't want to have an interface that puts all of this in front of your face when you just want to work on your labels or your reporting or setting up your program. So by breaking down what needs to happen in each of these workflows means that your interface is going to be a lot more clean. So here's an example of how Breeding Insight will be easy to use. And this is just a draft, so don't get too attached to it. But just as a welcome screen, it's welcoming back Liz, who yesterday, apparently she was editing and working on this data set from a field trial. And it's listed as in progress, so it's not been completed, so she knows that she can come back to it. But maybe, she's done with work. She doesn't want to work on that one today because it's exhausting, and she wants to think about it a little more. But then recently she was working on a different set of experiments, so maybe she wants to do that now or go to an older experiment. So you're going to see that it's got this clean, clutter-free interface, and it's going to be populated with the most recent activities that you were doing, because the chances are you're probably going to be doing it again. But if not, you'll have this portal that will take you to other workflows to, if you're at a transition stage, to start doing something else. Okay. Another example screen is if you were creating a new experiment. Instead of leaving it up to the breeder to know exactly where to start, the software is going to ask you, what data set do you want to use as a starting point? Do you want to redo another trial or pull from a trial, pull from your inventory, or pick from accessions? So it's going to be a very task-focused interface, and it's going to guide the breeder through the workflow. So even if they only do this create experiment once, 1 month out of the year, it's not going to have a very high relearning the next time they come back to it. And we really think this will make it a lot easier to adopt software and these types of tools into an existing breeding program that already isn't doing any genetics or is doing very little genetic work already. So having all of the data in one place and organized in a workflow setup means that that part of it is going to be a lot easier and more seamless for them to take up than having to learn the interface of 4 or 5 different systems and know when to use them and how to use them. So I want to talk in the last section just about completed and ongoing software development. So as I mentioned, these were our targeted integrations for our first platform. Again, we have other things that are not been included in this, but these are the big ones. So the first thing I want to talk about is the integration between FieldLook and Rebase via Rapi. That integration was done by Breeding Insight, and I have one comment before I go on and show that, is that even though we're showing it connected to Rebase, we've provided that software integration out to EIB, so Excellence in Breeding now has that, so they can use it with BMS or B4R or any other Rapi compliant database, phenomics database can be using the same integration. So, and that's one thing I didn't really remember to let everyone know, is that we're working in open source software and we're pushing out our products as an open source availability as well. Okay. So before I go on to this, what the integration looks like, I just want to spend like 3 slides talking about each one of these individual pieces of software or the in Rapi. I apologize if this is old news to you, but there's always somebody who maybe isn't quite aware of what Rebase is. So I just want to make sure everyone's on the same page. So I'm going to start with FieldLook. It's an Android app developed by Trevor Reif at KSU. It falls under the PhenoApps suite, and it's used by breeders to facilitate trial data collection, including photographs. It's meant to replace hard copy field books with digital media. It does allow you to do remote collection, and it's supposed to reduce transcription errors, although we haven't actually tested that. So that's something we'll be doing in Breeding Insight, is actually testing whether or not it does reduce transcription errors. And Rebase is a breeding management system with analytical capabilities developed by Lucas Mueller's group at BTI. And it's used by breeders to do a lot of things. So it manages seed lots and pedigrees, trials and methods. You can actually design your trials and create field layouts and maps. You can store your phenotypic data there, design tissue collection layouts using the coordinate app from PhenoApps. And it also can hold and use genomic genotypic data, so you can run genomic analyses like genomic selection. The last thing is Rapi. So I guess, it's probably not unfamiliar to many of you, but Rapi is just short for Breeding Application Programming Interface. And it's really just a protocol for getting different software to talk to each other quickly. But as a user, one should never have to see it or know any specifics about it. So the way that FieldLook and Rebase worked prior to then was through manual file transfer. So this user here would have to create an Excel file. You pull something out of Rebase, create an Excel file or a CSV file, and then upload it into FieldLook and then go out to the field. You can see that that's very tiresome and a lot of problems can go wrong with the template not being correct, or the transfer not being, it could be corrupted during that time period. It also is very difficult for some people to navigate the Android file structure on a tablet. So the preferred method really is to do this automated file transfer. So now we have the same user who's just interacting with 2 different databases through Rapi. They don't actually see it, and it makes the system feel like it's more holistic than 2 separate systems that are talking to each other. So this integration is available now. I just want to show some screenshots of what it looks like. So if you already have a Rebase instance with your trial data or your trial map inside, you can actually pull down that field or multiple fields into FieldLook through this integration. And in this example, we're pulling down a 2018 grape trial for downy mildew. It gives some description of what that file field is from Rebase, and it reads in the location plots and how many traits are coming in with that. We put that on our tablet. We go out and we collect field phenotypes based on using the app. And then when we're done, we're ready to export back. We can use this Rapi export function. And when you export it, it actually gives you a small report about what actually was done to the database when you exported. So in this case, we have collected 3 new observations. We synced 2 of them, and 1 of them was an edited one. So 2 were brand new, but 1 we edited. So it's a new observation, but it's actually overwriting an older one that maybe we felt wasn't correctly accurate or wasn't correctly put in. It also tells you if you've skipped any observations. So if you've skipped a whole trait, or if you've skipped a particular plot or range, it will give you that information too. So as I mentioned, this is now available in FieldLook version 4.1. And if you've been using an older version of FieldLook but want to try the integration with your Rapi compliant database, you can go to the Google Play store and actually download the new version with this integration. Our next software priorities are really to focus on some of the things we think are real problems for getting programs set up. And one of them is ontology creation and management of traits. This has been for us probably the single biggest delay that we've experienced. It holds everything else up for us. And if we're going to be pushing Breeding Insight software out to more and more crops over time within the ARS or even with public groups, then we really want to make this a lot less painful and a lot faster. So finding a microservice tool or creating a microservice tool to do this will be very beneficial. And we're still also working on our flowcharting steps and decision points in our program management. But soon we'll be working on the historical data load microservices. So this is for us, we find a lot of our programs some of our programs have been running for more than 50 years over multiple people's tenures, and the data is in all sorts of different formats. So we want to make it easier to get those historical data loaded into the database to start using that data and securing it, because right now some of it's in Excel, some of it is in paper format, and if that data is important and we want to secure it, we need to make that happen through a digital media. And we're also working on inventory management as a microservice. So currently Rebase allows you to keep track of seeds, but our breeders have a lot of other needs other than just managing seed. They need to manage tissue culture stocks, how long they've been there, when they need to be replaced, who they've sent them out to. Some of our groups share pollen, and being able to say, well, this pollen is available. If anybody would like it, just let me know, I'll send it out to you. Tubers. So having an inventory that's actually living instead of just an Excel file that if you forget to say you've taken seed out of this particular packet, that the next time you go there, you may find that it's gone and it's too late because you've already decided you wanted to put that out as a parent and you no longer have that seed available. So a lot of what we can do immediately for our breeders is help them manage their traits, manage their historical data to ensure that it is secure and usable, and really help them manage their inventory better. And that's our first goals right there, is to really help shore up those programs from the ground up. But coming this fall, we're going to be having a 1-week training course on deploying and using the Breeding Insight software wherever that stands at that point, and also the integration with using FieldLook 4.1 or later for data collection. And so if you're interested in, or you have a breeder that you work with that might be interested, I would recommend that they drop us an email and let us know that they want to be considered. So when we get our program out and start vetting people to come in and opening the enrollment, that we can let them know to look out for it and send them an email for that. So with that, I just want to thank you all for listening to me talk. I'm sorry, I had the flu last week and I gave a talk yesterday and I'm losing my voice now, so I apologize for the scratchy nature of my voice, but I do want to thank you all for being here, and also for my team. I have an amazing staff team that works with me, and none of this would be possible without them. They're all amazing people for their own rights. And Ed Buckler and John Luke are our PIs, and they have been just phenomenal support and technical resources for us. Our funding comes from the USDA Agricultural Research Service, and we're hosted at Cornell. So with that, I'll take any questions.
All right, thank you, Moira. This is a great talk. Does anybody have any questions so far?
I have a question. This is Effie. You mentioned that you have a microservice for ontology terms.
We're building one.
Oh, it's in progress. Okay.
Yeah.
Okay. And so would that allow someone to, check to see if a term is legitimate, or to look up do a little search on their own website?
Yeah, I think that could be a possibility. It's probably not the primary first release need. I think the first release need is to help people who are just using Excel to put things to document how they're actually doing their trait collection. So the primary use case would be they would actually build up their traits for their program that are important, and then we may have, it's not been fully spec'd out to do this, but there may be an opportunity to say, is this trait available already from crop ontology, if they want to do that. But a lot of our programs are either using crop ontologies already but don't realize it, or are so insulated, or they just don't really have any collaborators to work with to come up with a global ontology. So we want to just make it easy to get their data into the database. That's the primary reason for helping make sure that their traits that they care about and they have data already for are into the Rebase system, because you can't do anything with Rebase unless you have an ontology built.
Okay, so you would be typing into the crop ontology, plant ontology?
Well, the crop ontology holds flat files, at least the way we've seen it. It holds flat files for individual crops. But those ontologies, when they're held in a flat file, they don't mature. They just they're already outdated. And so that's our concern, is that we have breeders that do want to try new traits out,
Right.
but without having to go through a huge bureaucratic ontology review to do that. They may want to just check for one season if this trait has any heritability, and they don't want to formalize that trait until they're sure it's going to be worth it. So I'm trying to be as flexible for our breeders to make it as easy as possible. But if we have groups like the sweet potato group that says, no, we are working directly with the global sweet potato ontology, we are working with their curator for ontology to make sure that the new traits that get added don't conflict with anything they already have.
Okay, so sort of community by community.
Exactly. Yep.
Okay.
And the grape community has a lot of proprietary grape traits that they do not want to share with industry, because that is their only proprietary edge in many ways. So we want to give them the flexibility to keep some things in their own program isolated, if that's useful to them.
Okay, thank you.
You're welcome.
Hello, is the training course only for those 5 crops?
No, we're going to be opening it up to anyone. So it could be I don't know how we'll do the vetting, if it'll be first come first serve or how we'll do it, but it will be open to public institution breeders. It will be open to company breeders if they want to, deploy this on their own system, or a small seed company wants to. And it can be for any ARS person who's not in the program now. In fact, it's mostly designed for anybody who's not in BI currently, because we're doing the work for those 5 groups.
Okay, thanks.
You're welcome.
Any other questions? I was curious, Maura. So it seems like it's what you're trying to do is tie a lot of different complex software and data pieces together for crops that have not had that privilege or that previously. And so it seems to me that you're probably kind of bumping against use cases that had not been considered before in the development of these software platforms. Like, I don't know if FieldLook can handle trout trait data, So do you have any stories about where you think the most growth has occurred based on Breeding Insights development efforts to be more inclusive, I guess, of different crops?
That's a fantastic question, and it comes back to us really trying to do this with an agnostic mindset. The biggest thing that we've noticed is that most of the genomic databases don't deal with polyploids very well. They're still working on a diploid model, and we already know that at least for autopolyploids, the dosage calling is a problem. We lose a lot of our signal when we don't have all 5 classes represented, allelic classes. So by treating white potato or sweet potato as a diploid, you're really just doing a good job of estimating the AABB calls, but not doing a very good job with the majority of what's in between that. So that's been a surprise, was that the way data is still being collected, it's not able to really work with local haplotypes. It's not able to hold a bunch of, even like 2 or 3 SNPs or markers as a locus and use that as assigning alleles. Like in grape, we have 900 alleles for some of our loci. It's ridiculous. So if we can't use the haplotype information, then we're now treating it as a single SNP by single SNP. And that's been a problem. And it's something that we haven't resolved how to deal with. So we're working with the breeding, the Excellence in Breeding Polyploid Working Group to develop a standard for what tools and data management and VCF files should look like for polyploids that deals with all of them in a very agnostic way with less heuristics, where you don't have to know exactly the ploidy, but the data can be informative as to what that ploidy is. So we haven't solved that problem, but that one is a problem. Another good case is that FieldLook doesn't really work for trout, as you mentioned, but if they could collect their animal welfare data, which right now is on pen and paper and whiteboards outside of the tanks, the room tanks, with no way to notify somebody if there's an issue, if we could get that in a FieldLook type app where they could actually go in, they log in, they date stamp when they check the salinity, they date stamp the temperature check, and then they sign off on it, and they do their 5 things they need to do and they date stamp it. The next person who comes up can see it, and if there is a problem, they can flag it right away, and that will go to somebody to notify them immediately. The salinity in tank 5 is too low and it needs to be dealt with, and then signed off that it was dealt with. So that will help a lot with the reporting for that, which right now comes down to looking at watermarked pages that are all wrinkly and paper, in pencil and smeared, and having to type that in every couple of months. And it's a pretty ungratifying job for whoever has to sit down and do that. But that's what they're required to do for reporting back out, to show that they have good animal welfare standards and they're abiding to all the regulations for ethical reasons. So those have been 2 of the biggest surprises for us. And so we may not integrate something like exactly like FieldLook into the trout program, but we may revamp it as something that's more along the metadata collection for animal welfare. And the last thing that was kind of surprising is that we were approached by a group called Ag Voice, and they've been testing several different lexicons for voice phenotyping. And they have some videos, you can look them up, they're quite interesting. They use a SWAT microphone that has set 40 decibel dampening external sound, so you can use it in a windy cornfield. And they're able to just hands-free start phenotyping by talking to it, natural language. And when you look at a technology like that, you start thinking about where could you use that? Do you need a barcode scanner if you could just read off the label? So I think keeping in touch with what technologies are coming along and how they're developing and vetting those technologies means that we can take a more holistic approach instead of saying, well, everyone uses FieldLook or no one does, or maybe it works for some traits, but the voice actually works great for strawberries, because they're not, when you're phenotyping strawberries, you're climbing around on the ground and you don't want to have pen and paper in the mud with you. If you could just talk to it, it would be a lot easier. So trying to find technologies that actually are natural, that adapt to the human part of the requirements instead of the other way.
That sounds really cool. Thank you.
You're welcome.
I
also want to let people know that if they have other questions after you leave here, 10 minutes down the line or 2 days, you feel free to email me anytime and I'll respond to your questions through email. So I'm a slow thinker sometimes, so I'm always the person that's like sending an email like 2 days later after I've thought about something. So I want to make sure people feel comfortable doing that too.
Yeah, I got it. I have another question that's maybe related to ag biodata. So the primary data that's being collected as part of these projects, as I understand it, it's not going to stay with Breeding Insights. It's going to be hosted by some of the partner projects that you mentioned, for example, Rebase or whatever the database for trout or other species would be, right?
Well, we're actually doing our own hosting of those. So it is going to be held by us, but we don't own that data. We're just securing it and making sure that the user access to that is guarded. So but the program manager or the breeder in charge has all of that data available to them, and they make the decision about release or not of that data. So we'll hold that, but at least right now, the way we've set it up. But in the future, they may be hosting on ARS sites themselves. So that would fall under their data management policies.
Okay, thanks.
Yep.
Any other questions? All right, well, Moira's offered for people to email her afterwards, so that's available. But let's all thank you, Moira. This was a really interesting and great talk about a really complex and useful platform. So thanks much, and we'll keep in touch. All right, thank you, everyone. Bye.
Thank you.
Bye, thanks.